My name is Doruk. I'm a research scientist at deCODE Genetics, and my work so far has consisted of the analysis of structural variants using long reads in the Icelandic population. And I'll be presenting you our paper published in Nature Genetics on long read sequencing of 3,622 Icelanders, provides insight into the role of structural variants in human diseases and other traits. That's our title. And we're fortunate enough to have our cover suggestion approved by them. Having a Icelandic lopapeysa, a wool sweater. And today I'll be focusing on one example on each of the human disease and the other traits that the title says. The first example I'll be focusing on will be SV's role in LDL cholesterol, and another one will be on the human height. So let me give a brief introduction on the structural variants. They can come in several flavors, ranging from deletions to novel or non-novel sequence insertions, such as tandem duplications, mobile element insertions, or interspersed duplications, but also other events such as inversions and translocations. And the key here is that generally the field has focused on variations of at least 50 base pairs or more. And in our work, we focused on mainly insertions or duplications and also deletion events, because they constitute the vast majority of of the SVs, structural variants. So to be able to call them, we use long read sequencing, and we use specifically Oxford nanopore technologies as sequencing data. And the way it works is basically as it's depicted in this cartoon. The double-stranded DNA comes in, split into single-strand via the motor protein, and passes through the nanopore where the ionic current is constantly monitored. We call this a squiggle, or signal level raw data, as it's seen in the picture above. And because the changes in the current is dependent on the composition of the molecule that is passing through the nanopore at the time, we're able to use base colors and being able to convert the squiggle into a DNA sequence, which we use for downstream analysis. So one may ask us, why are we looking at SVs and why using longer sequencing? What is the main reason for us doing this? For SV sites, although they are less in quantity compared to SNPs and indels, because they're much larger, they also have a higher probability to have a functional role. And cumulatively, they actually affect either similar or greater number of base pairs. And there has been a series of literature showing the role of SVs in several diseases or other phenotypes in human. And the question for why long-read sequencing, one would say it is mainly due to the much increased number of SVs that we're able to get through long reads. For example, again, literature shows that 20 to 30K SVs have been observed using enough sequence coverage per individual compared to 2 to 8K of SVs using shorter sequencing. That is 3 to 5-fold difference from whichever number you're looking at. But more importantly, long-read sequencing reads are able to map much more accurately than short-read sequencing data, and it actually gives us a much clearer identification power for especially SVs, and they also have a much higher likelihood of covering an entire SV using even a single read. But they come with a culprit, and that is high error rate. The SV analysis workflow we use in our paper is, again, as the cartoon depicts, we have our individuals for which we generated raw signal level data, and then we use our base colors to generate sequences, basically FASTA files. We apply our SV discovery pipeline to every single individual, which I'm going to talk next. And once we have a set of SV candidate from every individual, we merge them using an algorithm. Using a cluster algorithm called CAST. And from the 3,622 discovered individuals, we've been able to get 133,886 reliably genotyped merged SVs. So once we have the way we reach there is basically, once we generate this merged SV set, we genotype them both using long-read sequencing data and short-read sequencing data. For the long-read sequencing data, we use genotyper that we wrote for the study called LR-Color. And for the short read data genotyping, we used GraphTyper. It was a tool already developed at deCODE prior to this publication. And we used our entire set of 3,622 ONT reads for genotyping as well as discovery. And we used a higher number of short read sequencing data, 10,000 specifically, to just get higher power on imputation, which is basically the next step. Using the longer phase haplotypes here at deCODE, we have been able to impute them into a much larger set of individuals, 166,000. And through various steps of filtering, we have been able to generate a high confidence SV set. And among the 133,000, about 120,000 of them were well imputing. So the SV discovery pipeline that I mentioned here in the orange box is as follows. As soon as we receive the raw reads, we map them to the reference, just HE38, a linear reference using Minimap2. From this, we use sniffles to generate sensitive SV predictions. So on top of this, we're actually using an alternate allele ratio based pre-filtering because, again, the coverage at the long read data is not as uniform as one may accept. Therefore, we are using a minimum number of alternate allele supporting reads cutoff, which sniffles at the time had as a parameter. And then on top of that, we're using, as I said, an alternate allele ratio based filtering, which looks at the variant allele frequency at the very site. So calculates the coverage there and then calculates the variant allele frequency. From that, we also use a tool called Swiper to do SV breakpoint refinement using short read sequencing data if we can. In a number of situations, it is short read sequencing is not helpful, but in several other places it is. And when we have both of those data, we're finally passing them through a tool we call Squiggle SV filter. It's basically a fork of nanopolish. What we were mainly used in this is we're mainly interested in nanopolish was actually its sequence versus squiggle likelihood function. So you can we can basically ask whether for this particular squiggle is this sequence versus this other sequence more likely. So here we're testing whether the alternate sequence that includes the SV is more likely for the raw squiggle level data compared to reference sequence. So here is actually one place where we go back into the raw data due to mainly the problems that we had in the base color due to error rates. So this section is actually with the, again, the study has started 2 or 3 years ago, so back when the base color accuracies were far lower, and the level of filtering this that provides is decreasing, the better base colors are. And just as a small outlook on how much filtering we've been doing, so the red dots we were able to see in the first figure here are coming from the number of SVs from each individual using sniffles and sniffles alone in our using the sensitive parameters. So we're basically trying to detect whatever we can in order not to have, let's say, false negatives. From that, we use this pre-filtering, which is just looking at the variant allele frequency, which is the blue dots. So everything that's red falling down to the blue one, everything in between is filtered out. And then from there, we apply the third filter, squiggle SV filter, which is pushing it even lower. So again, as you see from the smaller section in the smaller figure atop of the first plot, individuals where we have a massive number of SV calls is successfully reduced down their SV count to a more sane number. Guessing from the plot, it's more like about 20,000 or so compared to 150,000. But again, in perhaps the vast majority of the data where the quality is already fine, it is the level of filtering is less. And with the most recent data that we've worked on, out of the total 85 million sniffles per filter, so the blue ones, SV calls, we attained about 90% of them. So here in numbers, about 10 million SVs have been filtered away. But then again, one must always think the scale of this work using this set of thousands of individuals, because even though true SV calls will cluster, will merge together, incorrect SV calls, assumedly coming from random places, will not. And 10 million SVs can quickly drain the data in a sea of false positives. So these set of strict filterings prevented us from having such problems. The other figure on the right-hand side that we show here is that on the x-axis we have a set of children for which we have parent data as well, where on the y-axis we see the percent of child SVs not found on any parent. So the lower this number, the better it is. It's basically a proxy for one may say Mendelian errors, and we're just looking at whether the SV in the child is existent in either of the parents or not. So not necessarily, we don't do any genotyping here or not, or anything similar. That's just existence versus non-existence. And here, although in a number of samples, the error reduction is quite huge, all the way down to from numbers that are like 25% down to 5%. We're generally, from an average of an error of 4.5%, we halve that using the row-level filtering as well. And then the next step that I also glossed over a bit was the SV merging. So in general, one hopes for situations like this, basically clear-cut SV clusters in which you can just cluster them by looking at the breakpoint proximity mainly. However, especially with the long reads and SVs coming from repeat regions, we have problems like this where merging SVs become a tougher problem than otherwise. But to our belief, there must be two conditions that are set for, let's say, clean SV clusters or SV groups. One is that for single SV, we would like its different representations to all be similar enough to each other. And again, for single SV, would also like its different representatives to be to all be divergent from other SVs representatives or representations. And here, for simplifying some definitions, similar, let's just say that is not divergent, or divergent means not similar. And let us say that similar here also means that a minimum of n% reciprocal overlap. So there is a simple algorithm that can achieve these conditions for us. It's called CAST, Cluster Affinity Search Technique, where we represent we form a graph where each node represents an SV and an edge represents the distance between two SVs measured as 1 minus the overlap rate, the minimum of the two. And here the idea is quite simple. We start from the vertex of maximal degree in the graph, and then while at close, this is mainly used for gene expression data, so we can substitute the word gene for SV here, or node. While at close SV, let's say I, not in our current cluster or clique C, or a distant SVI in our clique C exists. First, we find the nearest closed SVI not in the clique and add it into it. And next, we remove the farthest distant SV from our clique again. And then, while this has been finished, we add the our clique to our partition, remove the clique, remove everything that's attached to it, every edges, and we rerun this until until every single node is removed from our graph. And this is able to give our cliques to us. So SV merging within repeats, however, is slightly different of a story, because this approach is not entirely enough for SVs in repetitive regions. It is because it is particularly difficult for mappers to place the SV event correctly within repeat regions, and it's even more difficult when the error rate is around 10% or so. So it sort of becomes arbitrary in a way where the insertion or deletion events will be inserted within especially similar enough repeats happening. So as I can show you a cartoon here. Let's say this is, here every box shows a repeat element. And then let's say we want to insert a new one. The mapper could just insert it right where it shows here, or it could insert here as well. It's totally based on the error rate of the long read and how close enough, how similar enough those motifs are. So there may be some randomness in this event. And even if they maybe are the same SV, they will never get clustered with the approach I presented before. Likewise, deletions may delete those 2 bits in here or there for the very same events, and then again they will never cluster together. So we'll have this explosion of SVs in these regions. So our solution was actually quite a simple idea. We moved their start site to the very beginning of the 10 repeat region, and then any clustering will actually work given their lengths. From this point on, we just apply the same algorithm, and after the merging has finished, and after we have our independent SV cliques or SV clusters, we can assign them a new start point, say based on the majority start sites in any SV that's found within a repeat region, and represent it as a multi-allelic SV. So our first results regarding the comparison of SVs found in long-read sequencing versus short-read sequencing was as follows. What we did is that we first looked at our SVs that are greater than 10% allele frequency against GNOMAD SV, a recent large-scale SV study, basically. We used 10% just because we wanted to make sure that GNOMAD would also have those SVs. So we found out that about 40%, 42% of them were not found in the GNOMAD data. And when we look more closely, we see that 47.4% of the SVs we find within 10 repeat regions were not found in the GNOMAD, and 27.4% of SVs outside of TR regions were found. So clearly there's an advantage of calling SVs using long reads within 10 repeats compared to short reads. And when it comes to genotyping, again, we genotype with both of our tools, LR-Color and GraphTyper. Out of the 120,000 imputed SV alleles, about 77,000 of them only were imputed from long-read sequencing, whereas only about 4,000 of them were only imputed using short-read sequencing. So again, the difference in genotyping was also quite drastic here, mainly because generally SV genotyping algorithms have required the SV events to be quite exact, whereas we opted out for a less exact approach in longer genotyping. And again, about 74% and 38% of SV alleles within and outside TR regions could not be imputed using genotype calls from short read data. Another basically point for calling, or not only discovering, but also genotyping within LRS data being more powerful than genotyping and calling from short read data, which basically showed one of the main advantages of long read data to us. So some general characteristics of the merged SV set that we generated. So from the 133 KSVs, 75K were deletions, 55K were insertion events. About 3K of them we did not resolve whether they were insertions or deletions, but they were large alternative events, different from the reference. And we found about a median of 22,500 SVs per individual. Among them, 13K were insertions and 9.5K were deletions. Some more insertions. This was also in line with other long-read data, such as from PacBio sequencing. And when we look at the length distribution for our SVs, we see basically a similar pattern again observed elsewhere, where the number of SVs decreased quite rapidly with length, and with a number of exceptions, where at length 300, 2.5K, and 6K lengths, due to retrotransposable elements shown in the color black here, we have peaks happening. These are basically elements that are reinserted or removed, so that is why they're increasing in frequency. And we've been able to replicate this general behavior as well with long-read data. Other sets of observations were regarding SVs found within telomeres or elsewhere. So again, there were much more 10 repeat SVs. The blue line here shows 10 repeat SVs. The red line shows non-10 repeat SVs. And the y-axis, the x-axis here is the distance to telomere. Near telomeres, much more 10 repeat SVs exist. Again, this is also similar to observations elsewhere. And likewise, the number of variants decreased quite rapidly with the allele frequency to here, the y-axis is in log scale. And we also next were curious about where the low frequency variants were found, and what we can basically say using low frequency variants. So in general, such variants, rare alleles, rare variants, or low frequency variants, they generally imply that they're either selected against or they're simply younger. So in order to be able to have a say in this, we look at two measures, one being percent of rare alleles at various regions. The x-axis shows the different regions that we're looking at, such as coding exons, introns, CPG islands, female-male recombination hotspots alongside double-strand breaks, and also chrom-HMM chromatin state regions, such as promoter, enhancer, regulatory regions, and otherwise. And so, as I said, we're first looking at percent of rare alleles and also the overrepresentation or underrepresentation of SVs for time repeat and non-time repeat in these regions. So we observed in the beginning both an elevated fraction of rare alleles and we saw an underrepresentation of SVs within regions such as coding exons and regulatory regions such as promoters and enhancers. And that was an indicative of negative selection to us when we have both, again, high percentage of rare alleles and underrepresentation. But curiously, within recombination hotspots, although we observed an elevated rate of rare alleles, we also observed an overrepresentation of alleles there, and that was indicative for younger variants, just simply places where new SVs are born, let's say. And here comes our first association result with this work. This is also the first human disease side of our paper, where a rare deletion in deleting basically promoter and the first exon in PCSK9 associates with lower low-density lipoprotein LDL cluster levels. We found a deletion of 14,000 bases with an allele frequency of 0.037%, and it had an effect of 1 minus 1.3 SD with a p-value of 10 to minus 20. We had 72 carriers of this deletion and quite a high number of non-carriers, 96,000. And we observed basically this deletion with this variant, again, a variant that is found most in West Iceland. And here, however, PCSK9, it's a gene that encodes the protein PCSK9, and it has been a key regulator of the LDL cluster metabolism, and it has actually even been a target of cluster-lowering drugs that Amgen uses. So, although previous work has been done on PCSK9 with loss of function mutations, this SV associating with the very same phenotype that hasn't been shown, although expected, and we have shown it in our data. However, there are other approaches in finding such associations, and one would be to correlate the SVs with previously reported markers such as the GWAS catalog markers. And out of the 116,000 GWAS catalog markers, 11,194 of them were in strong LD, with the 5,238 SVs in our data set, and a subset of 34 high impact and 54 moderate impact SVs were actually more interesting because they were, again, they were high in moderate impact. They were overlapping exons or spliced regions, so there's a higher likelihood that they're actually having a function. And they were in strong LD with about 200 GWAS catalog variants. And even more interesting would be such SVs in strong LD with low impact GWAS catalog variants, where the SV may be more explanatory than the SNP or endo. Actually, one such example we found out when we're looking at the height phenotype in a locus in chromosome 15 in the gene ACAN. We found exactly one such case where a synonymous SNP is in high enough LD with an exonic SV. And here, the SNP has been previously reported, the synonymous SNP has been previously reported to associate strongly with height with a p-value of 10 to minus 10. And we have basically found, one may say, a tagging SV, although the tagging, in quotation marks, SV was a 285 base pairs deletion from an exon And again, quite a high R-square, 0.96. And our initial verdict, as soon as we saw that, was probably that it is the SV that is the more likely culprit compared to the synonymous SNP. But when we looked a bit more carefully to the same sites where we found the SVs, we actually saw that we found many other SVs with different sizes. And we quickly realized that this was actually a tandem repeat region. They're calling variable number tandem repeat regions, V and TR. And here in the plot below, we see in green, the coverage coming from GNOMAD data. Basically, this is short read coverage where it's almost absent in the tandem repeat region of this exon, because it's mainly difficult to use short reads with direct mapping in this region. And here we had found 11 alleles, actually, several insertion alleles and several deletion alleles, as the cartoon depicts here. And they followed allele frequencies follows. Again, most of them were just no change at all, like the reference, compared to reference, but quite a lot of individuals also had several different repeat motifs inserted or deleted. And the most extreme forms we found was an insertion of 4 motifs all the way down to a deletion of 14 motifs. And to our surprise, we actually saw here, this plot shows on the x-axis a change in motif number, and on the y-axis the effect in height. We saw a linear pattern, a linear relationship between the number of motifs carried and human height. The lower number of motifs carried in that region were, the shorter the individual was, and vice versa. And this basically suggested us that there was a causal role in this locus with association with the height. And here, when we did a association with respect to the length of the allele there as well, incorporating basically length polymorphism as opposed to using simply the 5 motifs removed as we did before, we observed a much stronger association. So from 10 to the minus 9 or 10, our power increased all the way up to 10 to the minus 18. And we have observed an effect of 0.016 SD per motif. So that's, let's say, so the most extreme forms are, let's say it's 4 to minus 14, 28, let's say about, 20 times 2 for individuals to be homozygous carriers of these mutations. So it would be 40 motifs of difference. And with this ST in height, that basically give us a difference of 4 centimeters in height from the 2 most extreme forms observed in Iceland. Of course, when one looks at different data from other populations, this effect size could even increase in case we have, more extreme deletions or insertions. And here, when we dig a little bit deeper on the biology, we actually learned that the tandem repeats in this region, in this exon, are places where chondroitin sulfate molecule CS is basically attaching. So those are CS attachment sites, and the more attachment sites one has, the more CS molecule can get attached to the protein. And CS is negatively charged, therefore it is expected to draw in water into where this gene is expressed, where actually the agricon protein is mostly abundantly found, which is cartilage. So one expects the cartilage to be larger or thicker in here. So we, of course, do not know whether this makes whether this increases human height due to cartilage thickness, such as like the discs on our back or elsewhere, or whether it just makes the bones larger. We have obviously not done that study, but this basically made us change our final verdict from the previous one, which is basically larger the protein, the taller the individual. Another observation was again from tandem repeat in the gene PRDM9, which is responsible for the recombination sites. Here I'm showing on the top panel Illumina mapping, on the bottom panel ONT mapping. Although it's quite erroneous, it is able to display us these large deletions that we observe here. So the human reference in the zinc finger domain of PRDM9, which is basically this 84 base pairs motif repeating 13 times. In this individual, we have just 9 copies, 9 zinc fingers, so 4 of them is deleted. So 4 times 84 is the size of this deletion here. And long reads are easily showing this to us. And in this domain as well, we had found a series of alleles, again with a somewhat decreased, or let's say non-steady coverage from the short read data, the green track here in the mid panel, basically in this plot. And we had found all the way up to 1, 2, or 4 zinc finger insertions, and 1, 4, and 5 zinc finger deletions in our data set. And all of such different zinc finger motif length alleles have had a strong association with the location of crossovers. So previously, people were looking at the variation in this site using either SNPs tagging some of these alleles, sometimes multiple of these alleles. So it was hard to directly ascertain the effect of each motif count individually. Or they were using gel data, but I mean, such approaches are not high throughput. Therefore, one cannot do such approaches in a genome-wide manner, whereas with long reads, we've been able to achieve that. So our conclusions mainly have been that we've been able to identify over 22.6 KSVs per individual, which has been 3 to 5 times more than those found in short-read sequencing data. And we've also shown that longer sequencing is more sensitive than short-read sequencing, detecting SVs across genome alongside genotyping as well for SVs, and that the advantage is most pronounced in repeat regions such as standard repeats. We've reported 5.2 KSVs in strong LD with variants in GWAS catalog that are associated with a disease or trait. And this very number has been an increase of over 2-fold from what we get using SVs using short-read data alone, such as GNOMAD SV. And both a higher number of SVs called from an individual and a higher number of GWAS catalog correlating SVs indicates basically us that longer sequencing can further understanding of disease mechanisms in human traits and other human traits with via SVs. And I'd like to thank all of our co-authors from deCODE Genetics, also from Free University of Berlin, the laboratory in Mjot, and the National University Hospital, also the accredited hospital. And I'd also like to thank Dr. Jared Simpson for his help while we're developing squiggle SV filter from Ontario Institute for Cancer Research. And I'd like to thank you for joining me in this talk. And I'll be happy to answer whichever questions you may have.
Okay, yes. Now it's an opportunity for everybody to ask any questions that you may have. And you can also post the questions here, or we can invite you to the stage to be able to ask a question. But while you think about the questions, I can start off. Mm-hmm. So first of all, very impressive study on population scale sequencing.
Thank you.
Using the long reads. And it's really good to see like a new reference panels being generated in Nordic genomics. So I noticed that in your study that you report about like almost 10% of GWAS catalog variants are in like high linkage disequilibrium.
Yeah.
Do you think there's a way to further improve that?
To increase this number, basically this 11,000.
Yeah.
Well, obviously, when one is able to detect, more SVs, there's always the higher chance of being able to improve that. Or when one is able to, find more SNPs or indels, there's also a chance to also improve that with whatever set of SVs that we have. Also, increasing the sample size would probably affect this. It may be the case that, although, yes, no, I may just want to falsify myself directly. I believe the GWAS catalog variants were generally high frequency, or maybe more than 1% or so. So that may not be that useful. But yeah, I would mainly say if we will be able to call more SVs on one hand and more SNPs and indels on the other hand, we could increase that number. But surely we have in our own data false negatives, a lot of them, I'm believing, and many of them would probably correlate with the GWAS catalog members as well. So But so it could be possible to increase that even further. And I mean, we've already shown an increase from short read data, which had also used a large numbers individual sequence, about 13 to 18,000 or so from GNOME.sv. So one may, increase number of SVs just like we did from short read, from the long read, and obviously achieve a higher percentage.
So the combination of both is the good approach.
I would think so.
Yeah. Okay, and talking about the long read data, can you remind us again, like when it comes to nanopore sequencing on promethine, what chemistry was used for this study?
Oh, that is a very good question. I do not remember this off the top of my head, but I should be able to go back at the paper and give you some information there. But
was it like R9.4 or something?
I would believe so. However, I also should say that this has been work from 2 years or over 2 years, so we have used different ones. And even though the chemistry has changed over time, we actually we tried our best to base call them with the most recent base callers of Guppy. We had actually rebase called from our first submission. We had used some individuals. Yes, we had some flow cells that were base called using albacore and or earlier versions of Guppy. And then to be able to have everything in sort of in sync with each other, we have been rebase called SVs using the most recent versions of Guppy as well. Even, by the way, and even if we did that, we still had two different versions, although quite similar versions of Guppy. We still had multiple versions of it, and I believe this is part of the nature of a large-scale study that goes over 2 years, where the technology is advancing quite rapidly. But I believe it is the case that it was 9.4, the vast majority of it.
Yeah, that sounds really good, because my next question was actually, is there a plan to repeat experiments or sequence individuals with improved latest chemistry? So I believe that is one of the factors that contributed to the effective detection of SVs in individuals. And then you were able to, for example, observe this super interesting repeat motif that you found. Okay, so I had a few more questions.
Sure.
So I was wondering about, like, after creating this impressive, reference panel set, is there a plan to perform something like a telomere to telomere assembly of the individuals or of the reference genomes, maybe?
We would like to also create our own assemblies. Those are within our plans as well. And although we did not use any ultra long reads, we believe their usage could actually be quite helpful in achieving that goal. And those are definitely among the plans. And I believe having ultra longer reads would also help us to find more data within those telomeres, basically. Although right now, this work is mainly focused on just mapping the individual long reads that we have, which does not include ultra long reads from ONT, just the linear reference. But yes, those are within our plans, and we'll be curious to see whether that can boost our signal and lead to more findings. That would be something I myself will be quite interested in as well. Especially for an SV perspective, because I mean, as I showed in one of the earlier slides here, let me try to find that. Here, yeah. Here, basically, as we see, the closer we get to telomeres, the number of SVs, either non-TR or TR, increases. Although with the TR SVs, it's a massive increase. And one of the interesting cases that we presented in this work was from a 10 repeat region as well. So that would be of high interest to us too.
I would really look forward to see if any of these sort of studies from the Icelandic genomes. Okay, so we had one question in the chat. There were approximately 3.7K structural variants found exclusively by shortened reads. In general, have you been able to detect why these structural variants were not identified by long read mapping, and if any of them were of great significance?
So maybe the section hasn't been entirely correctly understood. So those, when I presented that section, the 3.8 KSVs were, all the SVs that we find here, all the SV discoveries that we're making are from the long read. We have not done SV discovery from short reads in the study. We have only used short reads to genotype to see whether they basically, the carriers are heterozygous carriers, homozygous carriers, or not, or non-carriers. That is the only use of the short reads in the study. And 3.7 KSVs were imputed by the short reads, and they have not been well imputed using long reads, which means that we have been unsuccessful at reliably genotyping them using long reads, whereas we have been successfully genotyping them using short reads. So I would say that is mainly due to mistakes that we haven't gone through solving at the time with our long reads genotyper. But from our perspective, it was all right to be able to genotype it using one method or the other. But this is a good question. We haven't spent the time to actually understand why we haven't been able to genotype it correctly, or we haven't. It could also be the case where, you know, so yeah, here I don't think there's any end in speculation. One may say that, you know, maybe the representation of the SV was wrong, and we have been able to find some set of carriers where, you know, the cured alleles, haplotypes make sense, therefore it imputes well using short-read genotyping. Whereas, you know, because we have not been able to identify the SV correctly, the long-read genotype didn't work well, or we simply had a number of problems in the long-read genotyping in those regions, and therefore, you know, we haven't been able to have a good result there. Or perhaps the coverage in the long reads in that region was, you know, not good enough to be able to get a good genotyping data, whereas the coverage in short reads have been quite well enough because, you know, in general, we had about 17x in median long read coverage, whereas we had 30x short read coverage. So perhaps it was a region where, you know, genotyping was easy enough with short reads, and, you know, the extra coverage that we had there helped. So this is, there could be several reasons for that, and we haven't looked at because it was a short number, it was a small number. But this is, yes, something where we may dig deeper and fix our longer genotyper, or if there's nothing to fix there, understand, you know, what is the culprit. Thank you.
Thank you. I was wondering about when it comes to detection of structure variants within repeats. As I understand that using the short read, it's quite difficult because you have mapping problems and, you know, also error. But when it comes to like long reads, you also have this high error rate. What do you think, in your experience, could be improved when it comes to long read sequencing technology itself to be able to have the improved repeat detection?
So, I mean, let's start from, you know, where short reads are. Short reads basically are almost perfect reads, let's say, in general, containing more or less, you know, no errors, I may say. And but the genome is a bit problematic, the human genome, and it's got quite a lot of places that look like almost identical to each other, or entirely identical to each other. Therefore, it is, when the read length is about 150, although it's paired end, it's quite difficult to make sure that those reads are indeed coming from that region, from the reference. Whereas with the long reads, just because the reads are much larger, much longer, and, you know, as soon as you're able to breach the repeat size in the human genome, there's no more question of inaccuracy in mapping it. It will most likely map correctly. And, you know, one may say that when you use enough coverage, initial works of SV calling actually have used coverages all the way up to 60x. Our approach focused more on increasing the sample size, and, you know, with more or less lowish coverage, 10 to 15, or actually median towards the end was about 17x. So there, you know, the expectation would be that because the error would be random enough, you know, reads will correct each other. The other reads will basically, so long as you do not see the same error happening over and over again, one will basically understand this is not a signal, this is an error. So this has been the sort of the mindset, in my opinion, with the long reads. And obviously, when we started, our error rates were about, you know, 15%, then they reduced to about 10% or so. I believe in this work, it was quite to, you know, around 10-ish%, let's say. And now it's going down all the way down to 7 or 6 even, with the most recent base colors, actually, without even the new chemistry or new flow cells or anything. So, and it also looks like it is the case that reads are getting longer too. So obviously, the longer the reads are and the less erroneous they will map much more accurately. Actually, I would even go ahead and say that at this point, maybe more than the error rate, the length of the read would be the most important one. And the larger the reads are, the less ambiguous the mapping will be. And then, so long as one has generated enough coverage, one should be able to fix the error problem. And any value that keeps on appearing on either half or all the reads should more or less be a real variant there. And I'd say the maybe the emphasis in long reads should be focused on the length of the read rather than error. But it's obviously much easier for us to have much less errors, especially much less systematic errors. So we won't have to deal with artifacts and other problems.
Thank you so much for these very interesting answers.
You're welcome. Thank you for the questions as well.
Yes. Okay, well, I think it's time to wrap up now. We only have 10 minutes. So I will share my screen. If you're interested in giving a workshop, research, or career talk, you can visit this link to be able to sign up. And if you want to recommend a speaker that you think we should invite next, then please let us know at bit.ly/suggest speaker. You are also welcome to host the talk from the speaker you want us to invite next. Again, thank you for joining us. If you missed the introduction, one of the important things we talked about what is Nordic computational biology and how you can become a member. So for that, you can visit our Join Nordic Combio link to find out more about it, and also visit our website, nordic-combio.icbsc.org, to learn more about the organization.